The tumour microenvironment is important because, clinically important because it's a therapeutic target. So everyone imagines that cancer cells and leukemic cells just live the way they are and are immortalized, but in fact they interact with lots of other normal cells, and those interactions can help them grow or prevent them from growing. And if we can manipulate that therapeutically, then that's a really good target for new treatments. So that's why it's important and interesting. I think a good way of thinking how a practical example of how the microenvironment has led to new treatment is the B-cell receptor pathway inhibitors in the treatment of CLL and other B-cell malignancies. So probably one of the biggest advances in treating these diseases has been the development of drugs that block B-cell receptor signaling. And what we now know is that in CLL, for example, B-cell receptor signaling happens in the tissues through interaction undoubtedly with other cells. We're not quite sure what those interactions are, but antigen is stimulating the tumour cell. Normally B cells get stimulated by antigen, and CLL cells are no different. And the realization that you could block that by drugs that block the signaling pathway has been a major advance in treatment. The key challenge in understanding the microenvironment is actually how complex it is, but also how inaccessible it is. So people are well used to studying tumour cells in haematology by taking a blood sample, and that just tells you what's happening to the cells in the blood. But in CLL and lymphoma, the important interactions take place within the tissues, within lymph glands, within the bone marrow. And we don't routinely get access to tissues in the way that we do the blood. So research into what's going on in the microenvironment has lagged a long way behind. So we have an enormous amount to learn, predominantly because we don't have access to materials. So that is one of the major challenges.